Hello everyone, welcome back. Today I'm going to be talking about cell membrane, also known as plasma membrane. So here we see a picture of the cell membrane. So one of the first things you're going to see is going to be the phospholipid bilayer. And then we're going to see some structures also in the plasma membrane, such as glycoproteins. I'm going to be talking about that. Proteins, all right, channel proteins seen here, some integral proteins also seen here. So let's dive into let's talk about what a cell membrane is made up of. So it's made up of phospholipid bilayer, it's made up of membrane proteins, like I said earlier, cholesterol, and then we're going to see some glycocalyx. So when we're talking about the phospholipid bilayer, you first see the word bi for two, all right? So bilayer is going to be composed of the hydrophilic heads. I have the next slide, I'm going to be talking about that, the hydrophilic heads, and then the hydrophobic tails. When you hear the word hydrophilic, the word philia, like Philadelphia, the city of brotherly love. So we're going to see it facing water. Hydrophilic would mean water loving, towards the watery surface. The outside of the cell is made up of water. The inside of the cell is going to be made up of water. So we're going to see the hydrophilic heads facing the water. The other part of the phospholipid bilayer would be hydrophobic tails. So you see the word phobia in the word, and you're thinking about phobia, fear of something. So hydrophobia or hydrophobic, not loving water, they're going to shy or shield themselves away from the watery medium. So they're going to be facing inwards. And these purple thing right here that we're seeing are going to be the membrane proteins, all right? And they come in different shapes and sizes. They're also known as kind of like a gatekeeper. There are going to be two types of them, the integral proteins and the peripheral proteins. The integral proteins are going to be found within the membrane, while the peripheral will be on the surface, almost like the one sticking out from the surface right here. All right, and then the glycocalyx, we're going to see glycocalyx are going to be this green structures right here. You see the word glyco, you're thinking sugar. So these are going to be surface sugars, and I'm going to be talking about their importance and actually like a clinical significance of their clinical significance. And I cannot forget cholesterol. Cholesterol are these yellow structures you're seeing right here, embedded in the cell membrane, and they help to keep the cell membrane stable. They give it further stability. All right. Okay. So first of all, what's going to be the function or functions of the plasma membrane, because it has more than one function? So first, it serves as a physical barrier. All right, it's a way for a cell to be separated from the cytoplasm, from extracellular fluids, which we call the ECF. And it also has a capability, selective permeability rather. So this is a way that it controls entry and exit of materials. Want to be able to regulate what goes in, what goes out. You would not want all your sodium just rushing into the cell, or your water. You want to be able to control, okay, all right, running a little bit low on sugar. We want some sugar to enter the cell. So we're going to have that selective permeability. So stuff like ions and nutrients are going to enter. We can have waste eliminated, some cellular products being released. Another function would be communication. Being able to communicate for one cell to be able to communicate with another. I'm going to be talking about this. Cell recognition also, all right? Protection, all right? The cell membrane gives the cell protection, and also structural support, anchoring the cells and tissues together. So here is a picture of the phospholipid bilayer, a close-up picture. You can see here the hydrophilic head, which is polar, all right? And then this would be the tails. This two squiggly things would be the tails. And when we take a look here, you can see the tails are inside. They're hidden away from the watery medium of the cytoplasm inside the cell, and the watery medium outside the cell. So let's talk about integral proteins, all right? So these are the ones I mentioned that are firmly inserted into the membrane. They often serve as transport proteins. These are going to be the channels, the carriers, the receptors like we're seeing here. We're seeing a chemical messenger like adrenaline, also known as epinephrine. This is how it's going to, cause, bind to a certain receptor. And then we can see a channel, all right? So let's assume these are solutes. This is a way they're going to be able to enter the cell. I talked about these proteins also. They also serve as anchoring proteins, stabilizing the cell membrane. These receptor proteins are going to be the ones that will bind in response to ions and hormones. And I talked about channels, regulation of water flow. We will want to control the amount of water that's exiting or entering the cell membrane. So for the peripheral proteins, these are going to be the ones that are loosely attached to the cell membrane, loosely attached to integral proteins, and their functions will include enzymatic actions, motor proteins, cell-to-cell recognition or connection. We're seeing this anchoring here, cell-to-cell connection, also here. So glycocalyx, all right, glycocalyx, these are sugars, carbohydrates. We see them sticking out here. They're the purple, all right. And sometimes they are attached, these sugar groups are attached to lipids. If they're attached to lipids, we call them glycolipids. If they're attached to proteins, we call them glycoproteins. And what they do is they are markers for cell-to-cell recognition. This actually allows your immune system to be able to recognize self from non-self, so that your body is not attacking itself. You wake up one day and your body's like, hey, what's this thyroid gland doing here? Let's just attack and kill it. So this is the reason why that's not happening, because of structures like the glycocalyx. So I already said I'm going to talk about a clinical case about how even, a small structure like this is important. So the glycocalyx of some cancer cells can change so quickly that the immune system is not even able to recognize it as cancerous. So, because sometimes, questions get asked, how are cancer cells bypassing this whole process? All right. So it's because the glycocalyx changes so quickly that the body or immune system is not able to recognize that, oh, this is not a good cell. All right, and this mutated cell will then will not be destroyed by the immune cells, which gives them more ammunition. They can replicate as much as they want. They're basically bypassing the what I like to call checkpoint, and we see that in the case of colorectal cancer. So what we're seeing here would be colon cancer. So this is, for example, how colon cancer, occurs or is formed. So I hope, you're able to learn a lot in this short video, and I hope to see you again in my next video. Okay. All right. Thank you. Bye.